Okay, it's gone 2 o'clock. Let's start. So good afternoon, everyone. Welcome to the last installment of the May Mini-school. Yeah, and I don't need to introduce you to the speaker. Betony, welcome again. Thank you for 3 very nice lectures, and we're looking forward to another very interesting lecture. Yeah, so Betony, please. And I think everybody knows the rules of the game. If you have questions, please use the Q&A, or at the end of the talk, raise your hand and we can give you upgrade you to panelists to ask the question. Yeah. Betony, please. Thank you very much.
Thanks so much, and thanks to everyone in the audience for coming today to this 4th lecture and final lecture of the mini-school on quantum biology. I hope it's been a useful mini-school. It's been quite broad, more than going into specifics, but hopefully it's given you some idea of where you might find more specific resources. The first 2 lectures, we went over some basic ideas about quantum biology from the physics and biology point of view. And we also looked at specific biological functions that could use quantum effects, such as coherent entanglement, spin chemistry, and tunneling. And last week, I went into more detail for one of these examples to outline the process of solving a basic quantum biology problem. Last week, we looked at spin biology, specifically whether quantum effects play a role in the nerves and brain. And this week, I want to focus on a different quantum effect, which is tunneling. And we're going to focus on tunneling in the specific biological context of infection by the SARS-CoV-2 virus, which is the virus that causes COVID. Before we get into the specifics of this example, I'd like to do a small recap of what we've covered so far. Over the past few lectures, we have looked at a number of different quantum effects, as well as many different biological contexts in which these effects may be playing a role. For example, we looked at coherent energy transfer processes in which energy is passed between light reactive molecules known as chromophores in a delocalized rather than one-by-one classical hopping manner. This coherent energy transfer has been investigated in a variety of different biological contexts. The most well-known of these is photosynthesis, but we also looked at how coherent energy transfer might take place between tryptophan chromophores and microtubules, and whether this is implicated in brain processing and anesthetics. We also looked at spin biology, where quantum spin states can have an effect on biochemistry and the role that entanglement may play in this. And I briefly mentioned what biological functions this may lead to, including magnetoreception, reactive oxygen species balance, isotope effects in xenon anesthesia, and lithium treatment of bipolar disorder, magnetic field effects on the circadian clock, and hypermagnetic field effects on neurogenesis and microtubule assembly. Nuclear spin biology might also play a role in the brain, which I looked at in some detail last time. So today, the quantum effect we'll be looking at in more detail is quantum tunneling. Tunneling was actually the first quantum effect to be experimentally observed in biological systems. And I have briefly mentioned in introductory lectures electron and proton tunneling in different biological contexts, such as enzyme function, olfaction, and neurotransmission. And today we're going to look in more detail at electron tunneling in viral infection by the SARS-CoV-2 virus. And you may wonder why I put the quotes around the receptors in the title of this slide, and I will explain that in more detail in the next few slides. Before I get into that, a quick recap of what I mean by quantum tunneling, which is a phenomenon in which a particle can pass through a potential energy barrier, even if it doesn't have the energy to overcome it classically. This happens because particles in quantum theory are functions which can extend into and beyond classically forbidden regions. So although the probability of finding the particle inside the barrier is low, it's not zero. So meaning there's some chance it can be found on the other side of the barrier. Quantum tunneling is essential in many physical processes such as nuclear fusion in stars, alpha decay, and many modern technologies like scanning tunneling microscopes. So I introduced the concept of ligands and receptors in lecture one, receptors are typically proteins located on the surface or inside cells, and they initiate biological responses when they are selectively bound to activating molecules. And these specific activating molecules that bind with receptors are often called ligands. And this interaction is fundamental to cell communication and signal transduction. It allows cells to respond to changes in their environment. Ligands can be an assortment of things. They can be hormones, neurotransmitters, drugs, or other signaling molecules. A major class of receptors is G protein coupled receptors, and you might have heard me talk about GPCRs. These are transmembrane transmembrane receptors, which means they span the cell membrane and they activate internal G proteins when they are bound by a ligand. And they are implicated in a wide range of physiological processing, including vision, smell, mood regulation, and immune response. Other types of receptors include ion channel receptors, enzyme receptors, and intracellular receptors. And they each play really critical roles in mediating signals that can control things as various as gene expression and muscle contraction. But in biology, the term receptor can be used very specifically, for example, these GPCRs, but more generally, all biological activities involve interactions between constituent reactants that might be thought of as involving a receptor and a ligand, with the more complex reactant labeled the receptor, so like an enzyme or a receptor in synapse or immunoglobulin, as opposed to the simpler molecule, which is then labeled the ligand. So that would be like antigen or neurotransmitter. I like to think of this as a sort of controlled catalysis that results from these binding specific reactions, which makes receptor ligand function absolutely central to biology and really a really interesting topic. So how exactly do ligands and receptors recognize each other? Well, ligand receptor interactions have to a large degree in history been explained using this mechanism called the lock and key mechanism, which relies on the shape of different molecules. The lock and key mechanism is a model used in biology to explain the specificity of molecular interactions, particularly between receptors and their ligands. In this model, the lock is the receptor and the key is the specific molecule that binds to it. And just like a lock fits precisely into a key fits precisely into a specific lock, only the correctly shaped molecule can bind to the active sites of the receptor, which triggers the biological response. And this specificity ensures that biochemical reactions can occur efficiently and with little errors. But there have been more nuanced models like the induced fit theory, which expands on this idea. But the lock and key is still a foundational concept for understanding molecular recognition and ligand receptor interactions in biological systems. Some of the pitfalls or shortcomings of the lock and key model are that it oversimplifies the complexity of molecular interactions. So binding often involves multiple forces or weak forces like hydrogen bonding or van der Waals forces, and the lock and key doesn't adequately represent these nuanced contributions. It also, to some extent, assumes that both receptor and ligand are rigid and unchanging in shape, but in reality, proteins are really flexible and dynamic, and they often undergo conformational changes when they are binding. It also doesn't completely explain the broad specificity of binding. Some receptors can interact with a range of different ligands, which contradicts the single key for single lock theory. It also doesn't explain allosteric effects. Allosteric effects are the phenomenon where binding happens at one site on an enzyme and it affects activity at a different site. And its allostery is crucial in many biological processes. So very quickly, to explain the induced fit, it's just induced fit model is a refinement of the lock and key concept that explains how enzymes and receptors interact more realistically with their ligands. Unlike the lock and key model, which assumes a perfect fit from the start, the induced fit proposes that the receptor changes shape upon binding to the ligand. And this allows for a more precise fit. But while both of these mechanisms prioritize the shape or the conformation as being central to ligand receptor, there's another theory that suggests that ligands and receptors are less like puzzles fitting into each other based on shape, and they're more like plugs. Their binding causes some sort of electron transfer to happen. And one of the ways in which this electron transfer could happen is through vibration-assisted tunneling, which I'm going to look at in more detail now. So you've seen this slide before. One of the main arguments against quantum effects playing a role in biology is decoherence, which describes the loss of these interesting quantum effects that I've been telling you about. And if you've read any quantum biology article, you'll have heard this phrase that biology is too warm and wet and messy for quantum effects to survive very long or on relevant timescales. Because quantum effects are fragile. They are normally observed only at very low temperatures, not at the physiological temperatures that life takes place at. And because at higher temperatures, the environment around the quantum system is moving and interacting too much with the system, which ends up causing decoherence. So for example, an electron transfer in biology is happening within proteins, and all proteins are vibrating at various different frequencies. But then there's been some suggestion that actually this environment around a quantum system might be very helpful. And biology is an extraordinarily clever engineer. And I think one of the clearest examples of this, which I've mentioned before, but I really like this example, is how we see different colors. So in our eye, we have 3 types of cone cell, and cone cells allow us to see colors. And cone cells work when light activates a light-sensitive molecule or chromophore called retinal, causing an electron transfer. But retinal only absorbs, if retinal is just in solution, it only absorbs light at one specific frequency. So how does this explain that we can see light at different frequencies? And the trick, or the ace in nature's sleeve, is the protein, because the proteins that retinal is embedded in for the different wavelength, to get the different wavelengths of light, are all different. So the opsin proteins, they have the same chromophore, retinal, but they have different protein structures. And it's the protein environment that tunes the light absorption in a favorable way. So perhaps biology has evolved to harness the movements of proteins to facilitate rather than disrupt quantum effects. And the theory behind vibration-assisted tunneling in quantum biology is that specific protein vibrations or vibrational modes and ligands facilitate tunneling in their respective receptors. In this process, the vibrations of relevant molecules help align the energies of donor and acceptor states, enabling quantum tunneling to occur more efficiently. And tunneling has been implicated in several biological, or suggested to be implicated in several biological processes, including enzyme catalysis, olfaction, and photosynthesis. For example, in certain enzymatic reactions, proton or electron transfer is too quick to be explained just by classical over-the-barrier activation, and suggests the involvement of tunneling that is facilitated by protein vibrations. And this vibration-assisted tunneling allows for highly specific and efficient biochemical reactions, and often at rates that defy classical expectations. Vibration-assisted electron tunneling in the context of receptors is most often talked about in the biological context of olfaction, which is the sense of smell. In this example, the quantum particles doing the tunneling are electrons. As I've already explained, many biological interactions involve this receptor and a molecule that binds to and activates the receptor, called a ligand. And olfactory receptor-ligand interactions are conventionally explained using shape-dependent models like lock and key mechanism. In contrast to this, the vibrational theory of olfaction, which was formalized by the biophysicist Luca Turin, suggests that the vibrational spectrum of activates electron transfer in its olfactory receptor. But what evidence is there for these vibrational theory of olfaction? Well, the standard experimental technique for testing the theory is by selective deuteration, which means replacing the hydrogen with its isotope deuterium deuterium has one extra neutron in its nucleus, which changes the mass and thus the vibrational spectra without changing the shape of the molecule. So it shouldn't affect the lock and key molecule. That shouldn't affect the lock and key mechanism, but deuterated small molecules, and deuterated, if it's not affecting the lock and key mechanism, then deuterated molecules should smell the same. But experiments have shown that fruit flies appear to be able to differentiate between deuterated varieties of smell molecules. This isn't definitive evidence because insect olfactory receptors differ from mammalian G protein related olfactory receptors. So it might not be that the results are generalizable. There's also certain species of fish like lake whitefish and the American cockroach have also demonstrated an ability to differentiate between isotopes of different amino acids and pheromones. On the other hand, there have been a number of studies that show that receptors cannot differentiate between isotopes, and the review article at the top right is a good one to read if you want to look at the current consensus on vibrational theories of olfaction. And there's also been some suggestion that olfaction uses a combination of shape and vibration, and this is called the swipe card model. And I included the paper on the bottom right because although taste is related to the sense of smell, there's been very little development of quantum theories of taste, despite the fact that humans can actually taste the difference between normal and deuterated water, which I think is pretty amazing because that's a very tiny change. A second biological function that may use electron tunneling that I've talked to you about before is nerve activation, or the binding of neurotransmitters to receptors on nerve cells. Neural communication is mediated by the release of neurotransmitters. When an action potential propagating along a neuron reaches the axon terminal, it triggers the release of neurotransmitters into the synaptic cleft, which is the gap between two neurons. These neurotransmitters then move across this synaptic cleft and they bind to special receptors on the adjacent neuron. And they open ion channels, and ions can now enter this adjacent nerve cell, change the membrane potential, and potentially generate an action potential in this adjacent nerve cell. And this is how a signal in nerves gets passed on. And the majority of receptors that bind to neurotransmitters are GPCRs, the receptors I've already mentioned, like olfactory receptors. So it comes as no surprise that researchers have tried to apply vibration assisted tunneling theories to neurotransmission as well. What success have they had with this application? Well, there is some theoretical evidence focusing on the neurotransmitter serotonin, which I talked to you about quite a bit last week, and because it is one of the fundamental neurotransmitters in the brain. And they looked at serotonin, and they looked at the vibrational spectra of serotonin and related molecules or ligands like LSD that activate the same receptors. So serotonin and LSD both activate the same receptor, but are to very different potencies with very different effects. And they compared them, and they showed that they share an inelastic electron tunneling spectral peak, and that the intensity of this peak can be used to predict the potency of the ligand, which is still quite an open question in pharmaceutical research. However, this unfortunately wasn't supported by a subsequent experiment which looked at selective deuterated ligands. In another couple of examples, computation other receptors and neurotransmitters such as histamine and adenosine did suggest that vibrational frequency was a factor that could play a role in the classification that bind to specific neuroreceptors. I think that a lot more experimental data definitely needs to be collected before any conclusions can be drawn. But one of the problems in these experiments is that it's very hard to get very pure samples, and any contaminant will change the vibrational modes. So that's apparently one of the difficulties in these experiments. Moving on to our example today, what other contexts might electron tunneling be important in biology? Well, It's hard to believe that the first COVID lockdown was half a decade ago. And during the pandemic, we had the idea to look at whether quantum effects play any role in viruses. And our approach was to look at whether there was any sort of receptor binding that we could apply tunneling theories to. So as I've already told you, ligand receptor interactions are pretty fundamental to all living systems. Ligands can be small molecules, such as drugs or pharmaceuticals, but they can also be proteins, such as the spike proteins that give the COVID virus its famous outline. The way in which this virus invades its host cell is by using the spike protein as a ligand to bind with receptors on the host cell. So in humans, angiotensin-converting enzyme 2, or ACE2, is a receptor that binds with and modulates angiotensin proteins, which are proteins that are implicated in things like blood pressure. But these receptors are also how the COVID virus invades a host cell. The coronavirus spike protein binds with transmembrane ACE2 receptors to access host cells. So angiotensin receptors are GPCRs, like olfactory receptors and serotonin receptors, which have been the main site of these tunneling theories. Angiotensin proteins are responsible for a host of actions. I've already mentioned blood pressure, but they mediate most vascular properties, such as dilation and contraction of blood vessels, cardiac health, and many other biological functions. But the receptor that the COVID spike protein actually binds with is not a GPCR, but it's a related enzyme. It's this angiotensin converting enzyme 2, or ACE2, that binds with and modifies angiotensin proteins. So if we want to apply this vibration tunneling theory, we needed to find a candidate for electron transfer in this receptor. So one of the primary objections, I'd say probably the primary objection raised against vibrational theories of olfaction is doubt around whether electron transfer actually happens in receptors. But recent research from researchers in the around vibration tunneling hypothesis has identified some possible candidate sites in GPCRs, at least, for electron transfer. The requirements for electron transfer in a receptor include some sort of donor molecule with the highest occupied molecular orbital, often called a HOMO, that is higher in energy than the lowest unoccupied molecular orbital, or the LUMO, of its associated acceptor molecule. And one way in which this can be achieved is if a metal ion, which can shift a LUMO down in energy, is bound to the receptor protein. In further research, a number of possible molecular wires, such as tryptophan molecules, have been identified in GPCRs to support electron transfer. And you will have heard about tryptophan before, and you will have heard about chromophores, which are light reactive molecules and are abundant in biological materials and are central to much of the electronic charge transfer research in quantum biology. And interestingly, or most interestingly to me, is that the end point of this electron transfer, regardless of the path taken, is suggested to be this highly conserved disulfide bond that is integral to receptor function. And you can see it in all of these three diagrams of different types of electron transfer. You can see it as that double S. Disulfide bonds are central to protein stability, but there's also growing evidence that they can act as a redox switch, the function of which has been implicated in signaling processes. And just how this works in transmembrane receptors like GPCRs is largely unexplored. So while the focus in quantum biology has been predominantly on GPCRs, what is interesting is that SARS-CoV-2 infectivity linked to redox sites in ACE2 receptors, and specifically a redox site that involves a disulfide site. And differences in the infectivity of SARS-CoV-2 among is an established fact without any clear explanation. But some researchers did an analysis of retrieved amino acid sequences, and they showed that spike proteins and ACE2 receptors are rich in these cysteine residues. And cysteine residues are conserved in various SARS-related coronaviruses, and they participate in intramolecular disulfide bonds. And very interestingly, infection-resistant animals lack a redox active disulfide in their ACE2 sequences. So with that background, let's get to solving our problem in more detail. But what exactly is this problem? We are interested in whether electron transfer in a receptor is facilitated by the vibrational modes of the spike protein that binds with it. So our system of interest has two parts, the electron in the receptor and the vibrational modes of the spike protein that binds with it and shifts the energy levels that the electron moves between. We are interested in whether the electron moves between these levels from donor level to an acceptor level. And in our very simple model, we only consider one vibrational mode in the spike protein. So in order to model this behavior, we need the Hamiltonians of both the electron as well as the spike protein, as well as how these interact. To simplify things, we model the receptor as a dimer, where the Hamiltonian is given by HR, and epsilon E, D, and epsilon A are the energy levels of the donor D and the acceptor A levels, and J describes the coupling between these levels and the likelihood of transition. The Hamiltonian of the ligand, which is in this case, the spike protein, is represented as a harmonic oscillator with frequencies omega associated with the protein. And the interaction between the receptor and the protein is given by the Hamiltonian at the bottom there, where the sum runs over the interaction of the protein with both the donor and the acceptor. But when we do our numerical simulation, we presume that the interaction of the spike protein with the acceptor is 0. The coupling strength between ligand, protein, and receptor is given by gamma, and A and A dagger are creation and annihilation operators associated with the spike protein vibrations. So the Hamiltonians, HR, HP, and HRP represent the system in our model of the SARS-CoV-2 receptor tunneling. So last week, we mainly looked at coherent dynamics of our system of interest, meaning we didn't consider the effects of the environment. And this week, we're going to include the environment interactions. So what do I mean by an environment in this context? Well, in addition to the receptor interacting with the spike protein, this receptor is a transmembrane receptor, so it is embedded in a membrane that it interacts with. So the full Hamiltonian has to include the membrane dynamics as well as the receptor interaction with this membrane. The membrane environment is also approximated as a harmonic oscillator with a similar interaction with the receptor. In this case, gamma E's represent the coupling between the receptor and its membrane environment. In order to differentiate this from the coupling to the spike protein, this coupling is taken to be much weaker than the spike protein coupling to the receptor. The Hamiltonians HRP, which is the interaction between the receptor and the spike protein, and the Hamiltonian HRE, which is the receptor's interaction with the environment, represent competing interactions. So HRP is essential for the receptor recognition of the spike protein, but HRE, on the other hand, it just describes undifferentiated coupling to all the many environmental vibronic modes in the vicinity of the receptor. The spike protein vibrations are, as I said, discriminated from the environment vibrations through their specific frequencies as well as the stronger coupling constants. And the sum runs over both the donor and acceptor, as well as all the possible environmental vibrations. Sorry, can you hear me okay? Because my next door neighbor is actually drilling very loudly.
Yeah, we can hear you, Betony.
Okay, that's good. Okay, so now we want to look at what happens to our system as it evolves in time. Does the electron move from the donor to the acceptor or not due to its various interactions with the spike protein and environment? We use an open quantum systems approach. If you remember from previous lectures, the Schrödinger's equation describes how ideal quantum systems evolve in time. And by ideal, I mean quantum systems that are isolated and at low temperatures and don't interact with their surroundings. But as we've heard, biological systems are necessarily interactive, never mind the fact that biology conventionally warm at temperatures. So in this context, the Schrödinger equation will not be an accurate description of how an electron, for example, behaves in the context of biology. For a more accurate description of the electron's use open quantum systems theory, which means the system's evolution is no longer purely unitary, and it can't be described just by the Schrödinger equation and a unitary evolution operator. Instead, the combined state of system and environment is evolved unitarily, and then we trace out the environment to look at the evolution of the system by itself. So in our open system, instead of the state vector and the Schrödinger equation, the state of our open system is often described by the density matrix, and its evolution follows more general equations such as master equations, which I talked about in lecture 1. A typical master equation for an open system will have a term describing the closed dynamics and this dissipated term, which describes the effects of the environment. So what does our specific master equation look like? We assume a weak coupling between the receptor and the membrane environment. And so we apply the Born-Markov approximation. You might remember that Markovian master equations neglect memory effects in the environment. So we are assuming that any information passed from the receptor to the membrane environment is lost. And the first part of our master equation describes the system of receptor and spike protein. And the second term describes the environmental effects, such as decoherence and dissipation, where that kappa is the coupling of donor and acceptor to the membrane environment. We assume the initial state of the dimer is at the donor site, and the vibronic mode is initially in the thermal state. How do we measure, though, whether the spike protein can actually assist this electron transfer? Well, we look at the difference between the maximum transfer probability with and without the spike protein vibrational mode. So without the spike protein, for a dimer or two-level system with a donor and acceptor separated by this energy epsilon D and epsilon A, with coupling strength measured by J, maximum probability is given by that equation J squared over J squared plus delta squared, where delta is related to the energy difference between the donor and acceptor levels. And the electron is most likely to be transferred when the donor and acceptor levels are equal and delta is 0. So to get the maximum probability, so that's without the vibronic mode. With the vibrational mode, we use the master equation that I've just showed you. So the difference between maximum transfer with and without this vibrational mode is plotted for a range of parameters. We estimated these parameters with respect to comparable biological contexts, for example, vibration-assisted tunneling in olfactory receptors. And the coupling between spike protein and receptor, as gamma, is plotted from weak to strong coupling. And we have assumed that this coupling only occurs between the spike protein and the donor level. So that is that there's no coupling between the spike protein and the acceptor level. So you will see on the graphs that this coupling strength that I'm about to show you, not these graphs, is plotted as a fraction of the vibronic frequency, which is why it is unitless. Average energy levels and level couplings are estimated with respect to redox processes and other biological systems. And then this coupling between levels, or J, is then plotted for a range of different values. The frequencies of the vibronic modes of the spike protein are taken from studies that investigate SARS-CoV-2 using Raman spectroscopy. And this includes the vibrational spectrum for both the spike 1 and spike 2 subunits, as well as the full spike protein and the receptor binding domain. And we have, I'm going to show you results, and where we used, we plotted results for 3 specific peaks that correspond in the spike protein to the amide-1, the amide-3, and the phenylalanine, which are all molecules in that spike protein. So just some results very quickly. Our results were plotted for 2 different vibrational modes of various parameter intervals. For this 690 inverse centimeter mode, the results are as follows. So figure A, B, C, and D represent different coupling strengths between the donor and acceptor level. So that's J, from weakly coupled to strongly coupled. The white regions on the graph represent parameter regions where vibronic modes do not enhance any electron transfer probability. The redder regions are where these modes have a negative effect on electron transfer probability, and bluer regions are where they enhance electron transfer probability. And you can see that they have a marked effect on transfer in very selective parameter regime, and this effect grows as coupling strength between levels grows. When the coupling is too weak, like in figure A, the vibronic mode has very little effect. But when the coupling between the levels is too strong, so figure D, the vibrational mode begins to have a negative effect. So this is for the other vibrational mode that we chose. Once again, the figures A, B, and C, and D represent different coupling strengths between the donor and acceptor levels, from weakly coupled to strongly coupled. This vibrational mode shows the same trend as the last one I showed you, with increased enhancement in transfer probability with coupling strength, but only up to a point before the vibrational mode acts against electron transfer. This frequency mode also shows transfer enhancement in two specific regions, as opposed to the last one where it only showed one region of enhancement. So the other two examples showed the effect of our proposed model over a range of biologically viable parameters. We were also interested in testing the model at the extremes of these parameters. So we chose the dimer coupling strength with the least favorable outcome and plotted the effects of vibrational coupling for very strong coupling to the spike protein vibrational mode. This has an unfavorable effect at very small dimer detuning, which means the difference in energy between the donor and acceptor. But at larger detuning, the effect becomes favorable, showing enhanced transfer probability in distincter regions, especially for higher frequencies of the vibrational mode. Those were just a sample of results, but what are the possible implications? Is there any practical point to this? Drugs are screened for use by their various properties, and this means that vibrational spectra could be a new way of identifying which drugs might usefully interact with which receptors. Receptors such as GPCRs are one of the main target of pharmaceutical interventions, and drugs and ligands can interact with a variety of receptors, or alternatively can interact in a new way, giving an off-label, what they call an off-label use for an old medicine. Various GPCR targeting drugs have been shown to have a protective effect against COVID. These include antidepressants such as Prozac, histamine receptor agonists, inhibitory agonists of various GPCRs, including receptors for serotonin, histamine, and acetylcholine, have also previously shown antiviral action against the Ebola virus. And the Ebola virus is similar to the COVID virus in that they both share a spike glycoprotein. It could be that understanding more about this could give us some insight into long COVID, which is where up to 20% of people who have had COVID still have a variety of different symptoms. And more generally, it could completely change how ligand receptor interactions are viewed in biology and medicine, alternatively promoting conductance or insulation, acting less like puzzles and more like plugs, as I've mentioned before. So if you've been in any of my other lectures, you'll know that I like to finish off with some more speculative ideas. So in this context, what are the wider implications of electron transfer in receptors? And you'll have seen this quote, I think, pretty much in every lecture about life being nothing more than an electron looking for a place to rest. And the origins of life is a question that continues to intrigue scientists across all disciplines. There's one hypothesis that life began with a simple form of metabolism, but that theory runs up against this problem that you need enzymes to catalyze metabolic fixation. But early metabolism didn't yet have access to complex proteins such as enzymes to act as catalysts. So other catalytic mechanisms have been suggested, with mineral surfactants offering that redox spark that was essential to this prebiotic chemistry. And I mentioned in my first lecture that one of these possible minerals was iron pyrite, otherwise known as fool's gold, making it so much more precious than gold, in my opinion. And there's also been some suggestion that the binding affinity of the ligands synthesized in this pre-enzymatic metabolism acted as an early version of natural selection. So if you had ligands that more strongly bound ligands, they would then be accumulated into further, the ligands that weren't strongly bound would just float off, while the ligands that were strongly bound would be accumulated into further catalytic reactions, and slowly this built up into the more complex biological materials that we see today. So ligand receptor binding is thus fundamental to the origins of life. And what also seems central is this redox activity in receptors, this foundational importance of electron transfer. So conductivity in biomolecules, particularly, I think, the selective conductivity, which is conferred by appropriate ligand receptor binding, is a powerful tool for understanding not only diverse phenomena such as pharmacological potency and viral infection, but also possibly origins of life. But to balance this speculation, what practical investigations can be done to further these ideas? Well, as I've hopefully got through to you today, a great deal of biology operates through the binding and activation of appropriate receptors using ligands that match these receptors. Many receptors consist of transmembrane proteins that are coupled to these specific ligands. GPC receptors, as you've heard, are the primary target of a majority of pharmaceuticals. And this binding is primarily described in terms of the shape-based mechanism, like the lock and key. But what this shape-based mechanism neglects to be able to predict, or is not so good at predicting, is they can predict what ligand will bind to a receptor, but they are less good at telling you to what degree or how potent a drug will be. Many drugs are also what they call receptor promiscuous, which means that they are developed to target one receptor for a specific disease, but they also partially activate a bunch of other receptors, which can also lead to things like side effects. And ligand-induced conductivity, on the other hand, might give us some answers to these questions about potency and efficacy of different pharmaceuticals. But what sort of an experiment could you do to investigate this? Well, this is a very nice experiment that was done in 2019, and it looked at scanning tunneling measurements of single proteins between electrodes in a natural aqueous environment. And they showed that the factor that controlled the measured conductance is the nature of what electrical contact there was to the protein. And that it turns out that the protein that was the specific ligand make highly selective electrical contacts. So they used this STM probe, and they did it specifically with an epitope connected to the probe. An epitope is a ligand or molecule that recognizes and binds to an antigen, because the antigen is the receptor. And they specifically investigated they investigated Ebola antigens, and they suggest that this could be used as a diagnostic tool for Ebola infection, because when those antigens, if you prepare the STM probe with an epitope that recognizes the Ebola antigen, and you put in a sample of aqueous solution that contains this antigen, you get a conductivity, whereas if it's not, you don't. So it's a very effective way for testing to see if you have that antigen in your solution. They mentioned technological possibilities of the technique, but they also conclude there doesn't seem to be any function for this differential protein conductivity in biological systems. But we would like to at least try to use this basic setup to test whether you can use protein conductivity as a way to classify the potency of different ligands and show how how conductivity plays a role in protein binding and even things like protein folding. So I'm going to finish off now, but before I do, I wanted to address one of the questions I often get asked, which is what is the usefulness of quantum biology research? I hope I've shown you just how useful it can be in some contexts, but to summarize, look more widely at possible research outcomes and what role Africa might actually play in this research. What I found really exciting about quantum biology is that it has the potential to be fundamentally useful in a number of ways. It might have an impact on how we harvest, manage, store, and amplify energy that the sun supplies, which is a particularly important question for Africa, given that this energy is an abundant resource. Quantum effects in photosynthesis primarily focus on how energy from the sun is transferred to electrons located within photosynthetic structures in plants. These electrons then move along an electron transport chain to a reaction center, and this electron transport in turn drives proton transfer across the membrane, resulting in proton gradients, a form of stored energy. This is then used to synthesize ATP, which is the main chemical energy in cellular processes. And as I've mentioned, there's been some suggestion that quantum coherence can facilitate and enhance this energy transfer. This interest has now extended to the potential use of quantum principles for improved solar cells, and the collective absorption and emission of energy in plants has also generated interest in developing quantum batteries and sunlight pumped lasers based on these principles. As you hopefully know by now, spin is the property of a particle that dictates how it will behave in a magnetic field. It's also been instrumental in the development of quantum biology, particularly on the topic of magnetic sensing. Spin chemistry was developed already in the 1960s to explain how magnetic fields can change the results of chemical reactions by flipping a spin and thus allowing or forbidding a chemical reaction. Not long after this, spin chemistry was extended to spin biology when it was suggested that the Earth's magnetic field can interact with pairs of electrons within proteins in the eyes of birds, changing the spin state and thus changing a chemical outcome with a biological function. And similar principles have been applied to developing incredibly sensitive magnetic sensors from diamonds. And while the radical pair mechanism depends on the spins of electrons, the example I talked to you last week about suggests that entangled nuclear spins could be responsible for entangled neural firing in the brain. And because nuclear spins can maintain quantum effects such as coherence for appreciably long lengths of time, they are ideal candidates for quantum information processing in the brain, but also perhaps in the development of quantum computers. And I mentioned last week, what I find really interesting is that the model for nuclear spin entanglement in the brain is very similar to the original proposal for the Kane quantum computer, and both of them rely on the spins of phosphorus nuclei. And finally, there's a great deal of emerging excitement about the role that quantum biology might play in the future of medicine. This is definitely not new. It was of interest over a century ago, and I had mentioned in my first lecture that the 1903 Nobel Prize for Medicine was actually awarded to Niels Finsen for the use of light therapy to treat skin tuberculosis. But light therapy is still not an accepted mainstream medicine in any appreciable way. And I suspect it is because the mechanism is not understood and the correct dosages remain elusive. For example, there was a study during the pandemic that showed that the use of red light greatly reduced lung inflammation in COVID if it was applied for, I think, 10 minutes a day, but the effect disappeared if the application was changed. And can you imagine in a continent where TB, for example, is a massive problem, the use of red light LEDs, which are really cheap, safe, and accessible to treat lung inflammation, could be revolutionary. But what is really critical is understanding both the mechanism and the dosage, which is where quantum biology comes in. It's also believed that red light therapy or photobiomodulation, which has been used to treat many different conditions from neurological damage to depression to wound healing, even acne, acts on mitochondria. And mitochondria, you might have heard me call them the power stations of the cell because they produce ATP. And the way they produce this is similar to the process of photosynthesis. Electrons move across along an electron transport chain to create a proton gradient. Although this process uses chemical energy rather than light energy, the protein complexes also contain these light-sensitive molecules, similar to those that are active in photosynthesis. So shining light on mitochondria could thus enhance the production of ATP and stimulate the health of the cell. And quantum biology has made great advances in understanding the electron transport chains of mitochondria. to consolidate, to develop and use light therapies. Mitochondria are also primary sources of reactive oxygen species. Reactive oxygen species are important signaling molecules I've already told you about in biological systems, but they can also cause inflammation, damage, and death to cells. And I'm sure everybody is familiar with the popular health promotion of antioxidants. But what is really interesting is that recent research showed that spin manipulation of reactive oxygen species can be used to promote stem cell growth with a view to treating cancer. The problem to be solved here again is that of proper dosage, where what is the precise field strength that is helpful rather than harmful, which is another thing in which quantum biology can have a real impact. And as you've heard today, quantum biology is also interested in how proteins such as the COVID spike protein or other ligands such as drugs and pharmaceuticals bind to receptors in the body. And this has been investigated in the context of all sorts of things, olfaction, neurotransmission, allostery, as well as the coronavirus. And a better understanding of this could lead to the repurposing and development of new pharmaceuticals. And finally, in addition to these concrete outcomes, quantum biology might contribute to generating fundamental new knowledge and answering big questions such as how life started or what is consciousness. Thanks so much to all of you for listening and attending. I really hope it has inspired some of you to look into doing some quantum biology research, as I think it's a really amazing field. And if you want to ask me any questions or have any discussions, you can contact me on either of those email.
Betony, again, thank you very much for another fantastic presentation. I see there are lots of compliments and claps flowing up the screen. We have a few questions in the Q&A and one raised hand. So I would suggest that maybe we give a look at the question in the Q&A while I allow Mohammed to ask his question in person just now. Can you
Hello?
see the question? Sorry, Mohammed. Let's do quickly the Q&A and then we come back to you. Sorry, you were quite quick in accepting my invitation to talk. There is a question by Charles Johnstone, and he said, "What criteria do you use to select the Hamiltonian of the system?"
Well, I guess it depends on what you're looking at it. So in the spin in the spin example last week, we're looking at all the interactions that the spins could have. So the spins could interact with the Zeeman effect. I mean, they could interact with the Earth's magnetic field, so that gave you the Zeeman effect. They could interact with the nuclei around, so that gave you the hyperfine effect. There are also other effects like the electrons interacting with each other that give you an exchange effect and a dipole effect. This week we were looking at we were looking at an electron moving from a donor to an acceptor level, so that gave us the Hamiltonian for the electron, but then we needed to include the vibrational mode as well. So it depends on what's your specific system is or the interactions that your system is having, I guess, if that answers your question.
I guess so, yeah. Betony, there's another question by Youssef El. Sorry if I pronounce the name wrong. And the question is it accurate to depict the behavior of the protein and the membrane as a harmonic oscillator, given that the system is adiabatic? I think, he says, and the environment is high entropic like this. It's more likely for the vibrational behavior to be anharmonic, especially that it is coupled with an electronic Hamiltonian.
I would say go ahead and model that one and see what you got. They are mainly pretty much all the quantum biology interactions that I've seen model proteins as harmonic oscillators with specific frequencies. But I haven't seen other examples of that. If you have any, I'd be very interested to hear about them.
And I guess modeling harmonically is the first step. one can always get more refined in the various models. Graham, you have a question. You want to ask it in person?
Yeah, I might as well. Hi. So my question was on like the master equation that you use. Like, Do you derive it microscopically, or do you just kind of postulate it like phenomenologically?
It was more phenomenologically.
Yeah, because I was guessing like, how do you get the rates in that equation?
Yeah.
Have no
And then
speculation or,
most of the parameters are taken from, most of the parameters have been taken from literature.
Okay.
There is a last question, Betony, in the Q&A. And another just came up. And it's about if there's anything on antibiotic resistance with respect to quantum biology.
Yeah, there is actually, and I think it's in TB. Not so much antibiotic resistance, but antibiotic use in TB. And if you want to look up, I think it's John Joe McFadden, who wrote pretty much seminal book on quantum biology called, can't remember what it's called. What is quantum biology? Anyway, look up. It's a book by John Joe McFadden and Jim Al-Khalili. But John Joe McFadden's he's a biologist, and his main field of research is on TB. And I know he has a student who is looking at I think antibiotics and that. But if you want to email me, I can give you the details. I can't remember it off the top of my head, but yes, I think there is. And it was also really interesting because I think there was some involvement in chirality as well, where the different chiralities of the antibiotic were affecting something. But I don't want to tell you something wrong. I do know there is some research into it. Yeah.
Excellent. Okay, the question, the QA keeps coming. There is one question by David Silverman. Hello, I'm working to the National Association of Science Writers. Oh, that's very interesting. To develop several quantum biology articles, in part inspired by this presentation, and hope to follow up with you. What is your advice for popularizers and science communicators approaching these subjects?
That's a great question, because I'd like to know the answer to.
We would like to know also.
I also work in science communication. I had some success with the physics world, but I think knowing someone at getting it published is the hardest part for me. But I know Omid has published just on Medium, so that might be also an option. I wasn't sure if your question referred to publishing or writing.
I don't know.
But also email me if you want more.
This sounds like an interesting discussion, and we will be very happy to engage with you, David, on this. You have Betony's email, and it's probably not too difficult to find mine, and maybe Omid can share my email address as well. I think we should give now the opportunity to the two raised hands to ask their questions, and maybe let's start with Muhammad.
Thank you for the presentation. I just wanted to ask, what is the motivation to select your to calculate the probability difference between the with virus and without virus, and what is the meaning of your negative probabilities, which is occurring at the highest coupling strength? Don't you think rate of the reaction is more appropriate physical parameters in that kind of reactions?
No, I think maximum probabilities is the rate. Maybe it would be another way of approaching it, but I think the maximum probability is a good way of seeing whether you can enhance. So if you have just a dimer system and you know that the maximum transfer probability will happen when those energy levels are equal, then it's a useful way to compare it with adding a vibronic mode and seeing, well, does it actually improve the transfer? So that was the first part of your question. What was the second part? Sorry, I've
Second part is related to the negative probabilities or difference of the negative probabilities at the highest.
Well, it wasn't really negative probabilities. It was just that it was less. It just decreased the probability of the transfer of the electron.
Okay.
Again, sorry, maybe I'm not answering your question, but I'm happy to email you. I've seen your paper on the topic too, where I think you
yeah.
refer to rates, but maybe we can, I didn't really understand, to be completely honest, your method. So I'm happy to chat about it offline.
Sure. Thank you.
Excellent. Okay, I think Youssef has a question. Youssef, please, you're welcome to unmute yourself. Okay, no questions. Okay, here we go.
I'm sorry. No, I was just unmuting. Can you hear me?
Yes, we can hear you.
Yes.
Well, honestly, it's not a question more than it is advice. I'm an undergraduate studying biology, so biology is something I'm more passionate about. It's not really my main field of research, but I tend to really believe in biomimicry and being inspired by biological materials, especially as someone working in soft mapping and polymers. So I just wanted advice on what mechanisms, materials to look into to use for my work. Like I was really interested in things like the mitochondria and the genetic genes. It inspired a lot of work.
Hi, sorry, your connection is, I didn't really hear the question so well. You were just wondering what contexts in biology might be on soft. So, yeah,
Yes.
sorry. So yes, sorry.
I was saying that I work in nanomaterials, so I tend to believe in biomimicry, using quantum biology and same mechanisms, same phenomena that are used to make biology or biomaterials in my work. So I was just asking your opinion. What
I'm so sorry, I really can't hear you. Do you want to send me an email?
It's okay.
Yeah, okay, sorry.
Yeah,
Unless Francesco, can you hear better?
No, we have also here big difficulty in deciphering what the message is coming through. But maybe we can liaise outside this meeting and maybe try another connection. Yep. Okay, but I think this brings us slowly to the end of this mini school. So Betony, thank you so much for a fantastic series of lectures, which were very inspirational and I think made very clear that there's lots to be done. So let's do it. Yeah. And thank you, a big thank you to all the participants. We will upload as soon as possible the video and the slides and everything. And and we are looking forward to see all of you in other mini schools. The next one starts next week. Excellent, Betony, thank you very much. I see lots of claps. Thank you very much. Thank you.
Bye.